antibody response is similar to what you get as Jason to trigger primary immune response. It will be slow, it will be brief, low amount of antibodies. You mostly make IgM antibodies, and the affinity of antibodies to the pathogen or the virulence antigens will be quite variable. But when you go for secondary immunization, that will mount secondary adaptive immune response, the response will be very quick because of memory T cells and B cells. And that immune response will last for weeks and months. In the case of coronavirus, antibody titer to stay protective for at least 6 to 9 months. The amount of antibodies which will be made will be very high, mostly IgG type antibodies. So you get the best fit antibodies based on exon shuffling. So that's first immunization and second immunization, no different from what we discussed under primary adaptive immune response and secondary adaptive immune response. So what are the vaccine components, antigenic preparations? As we said, that it can be living, so you can take the whole organism, and natural ones are used. Vaccinia virus, Volibacillus virus, etc. have been used historically, but nowadays this is very rare. Mostly you have to attenuate them. You need to make them weak, and there are several ways you can make them weak. Polio is a classical examples, measles, mumps. So one is to chemically treat the pathogen. So it cross-links the protein, so pathogenicity goes down, but their antigenicity, that is ability to mount immune response, still stays. And they are very good for vaccine development. Many times intact or microorganisms can be used, but they are not live, so that they can't cause infection. And that is the case with polio, rabies, influenza, hepatitis, pertussis, etc. You can also use small fragments, for example, pneumococcus and many meningococcus and Haemophilus. You just use capsular polysaccharides, and you have to keep giving these immunizations repeatedly because capsular polysaccharides won't generate very high quality and high affinity IgG type antibodies. But in the case of hepatitis B, you just need one antigen, which is good enough for antibody response that will last for several years. In the case of tetanus, of diphtheria, you just use the toxin material. And the other ones which are experimental, and now you can see that it is actually in clinical trial, and now people are taking it. Examples are mRNA-based medicine, vaccine, which is being used by Pfizer. The other recombinant DNA-based technologies are to load viruses, isn't it? Like adenovirus, which is the Chinese vaccines are being used. So adenoviral vectors, which forms the scaffold of flu virus, and then you can put your gene of interest, the virus will infect and keep churning out the spike protein of SARS-CoV-2. The alternative is to check heterologous adenoviral vectors, which Oxford group have done. So the one which is coming from your Serum Institute, Pune, where you use chimp adenoviral constructs, where you have cloned the spike protein, and that will give you the antibody response against SARS-CoV-2. So there are different approaches which are used depending on the virulence factors available to the scientists. It keeps on changing. For example, you can have vaccination with polio, okay, which is perfectly fine. But if you use live, intact, then weekend one, attenuated polio vaccine. So the mouth drop polio vaccine, which was recently campaigned in India, they are attenuated. But what is the precaution told to parents is that when you change the nappy of the baby, then make sure that you don't get in contact with the baby's poo or urine, because if mothers are immunocompromised, then those viruses can be infectious for the mother. So those are the aspects which are important to take into consideration where you go for oral vaccine or you go for the intradermal or intramuscular vaccines. So all those things are very important to consider. So what is the broad and global understanding? What are the main requirements for a vaccine when World Health Organization thinks that this is a perfect vaccine or this is near perfect vaccine? It should be effective. Everybody should be protected. 8 billion people should be protected. It should be safe, no side effects or minimal side effects. It should be stable. You have heard the story about Pfizer vaccine that needs minus 80 degree for maintenance. And if they don't use it by evening or next day, then they have to throw it. If it is thawed, that is not going to work in India. That's not going to work in most of the Asia and Africa and South American countries. So you also need vaccine containing stabilizers, which can be carried at even room temperature, at best 4 degrees centigrade. That's why these things matter, because you are talking about a treatment or prophylaxis which must be effective globally. And affordability, we have talked about. Every citizen should be able to get it, because it's just not good enough to vaccinate 50% people in the world, isn't it? 50% people can still catch the infection and still pass it on if the antibody levels go down, isn't it? So it's a huge logistic exercise to vaccinate the whole world. With live attenuated vaccines, how do you attenuate them? We talked about, these are some of the examples. The commonest thing is to take heterologous cells. In this case, a monkey cell has been taken and the pathogenic virus is being asked to infect it. The more time you infect, the virus keeps on changing, because this host cell is not designed to accept the virus, because it is a human virus. So there is a selection pressure by the cell. After several passages, that is several rounds of infection, this virus, as it is natural for most viruses, they will start to undergo changes because they are not able to do what they would like to do, that is to kill the cells. Since these are heterologous cells, for example, the monkey cells, they are not getting dead. So virus will infect, complete its life cycle and come out. But because of this selection pressure, they will undergo changes. And by the time there are several rounds of passages, the virus that is coming out here is very distinct from here. In the whole process, it will lose its virulence, that is its ability to cause infection and death of the infected cells, human infected cells. But because there have been subtle changes in the virulence factors or surface antigens, they are still good enough to mount or generate immune response in humans. So they are called live attenuated vaccines. There are lots of examples: polio, measles, mumps, rubella. Okay. Alternatives, you can treat them with formaldehyde. And if you do that, then they become non-infectious, but antigens are still there. And those antigens will be grabbed by dendritic cells, follicular dendritic cells, and that can lead to production of antibodies and production of memory B cells and T cells. And these are some of the examples. Most of them you'll find in the books. I'm just discussing the principles. If you pick up one toxin, which is very important for that pathogen, for example, Clostridium tetani for tetanus, you just inactivate the toxin. And in most cases, they are the most important virulence factors as well. So if you give one, then you are free for almost 10 years because the antibody response is so good. Same is true for Vibrio cholerae that causes cholera. You take B subunit of toxin, cholera toxin, and then you can find that the immune response is very good. So you just don't have to take the whole pathogen. You don't have to attenuate them. You don't have to inactivate them. You can just pick up one important virulence factor of the pathogen. As I said, you can pick up subunit, one protein or even one fragment of that protein. Hepatitis B virus is one example. Neisseria streptococcus, Haemophilus, the capsular polysaccharides can be used. So you don't have to use the whole pathogen. And in this case, in case of SARS-CoV-2, you know that you are using mostly spike protein. So these are called subunit vaccines. You are not using the whole organisms. You also need components which are added to vaccines so that they can potentiate the antigen presentation and clonal expansion. So that's why if you look at the tubes of many vaccine, if you have a chance, you'll see that they always had white precipitate, and that's because of the presence of inorganic salts. There are also delivery systems like liposomes, etc. So mRNA vaccine from Pfizer against COVID-19, they are trapped in multicellular, multilamellar liposomes or lipid-like structures. So they can be gobbled up by phagocytic cells efficiently. They can also last longer in circulation, and then they can be antigen presented to T cells. So all those things are tried. Many people use CpG island. So just to stimulate, you know that if dendritic cells see something non-self, then they will start to flag it as non-self, isn't it? So dendritic cells will start to express co-stimulatory molecule. So you add a number of things of bacterial origin to confuse immune system that this element needs to be presented more efficiently because it is non-self. So again, the aspect of innate immune system comes into play, and those adjuvants are designed to do that. So they add value to the antigenicity and immunogenicity of the vaccines. This varies from one country to another country, depending on where you are. But as you can see that within 2 months, 4 months, 6 months, 15, you go for DPT, diphtheria, tetanus, pertussis, polio vaccine, you can see, MMR vaccine. These are the regimens when to go to the clinic, and mothers will take you to clinic for vaccination. Pneumococcal conjugates, Haemophilus, hepatitis B. So it depending on different countries, they all change time to time, depending on the data collected. But you can see that you go back to clinic and it gets recorded in your red book, how many vaccinations you had, at what age. And this is all in anticipation that if child doesn't get these vaccines, the child is likely to get most of these infections. So the pathogens are there. The only thing is that we are ready for it because our memory B cells and T cells are waiting in the wings because they have been primed by prophylactic use of vaccines. Vaccines are also not given to everybody, and they fall under restricted group of vaccines. For example, tuberculosis. So BCG is given only to people in tropical countries. So, for example, in the UK, every citizen here doesn't have to get BCG vaccine unless they decide to travel to other countries, which are endemic countries. You know that BCG vaccination is good only as a child, but adult, it doesn't offer you protection against adult tuberculosis. Hepatitis B, if you are meant to work with blood, etc. So if you're a medical staff, nursing staff, or a research scientist, or if you're a drug addict, God forbid, then you need those vaccinations in preparation for that, so you don't get in contact. For example, people who go for mountaineering in different countries or more extravagant sports activities, and if there is an accident and they need blood donation, then you need to have hepatitis B vaccine so that any contaminated blood doesn't give you the infection. Rabies is post-exposure. So you already have antibodies. We call it passive immunization. I will talk about it in next slide. and so on and so forth. There are a number of vaccines which you need to take in preparation for traveling elsewhere. Elderly people get flu jabs every year, and looking at the immune response against SARS-CoV-2, because it lasts only 6 months or 9 months, and you expect that if it is not going to go away for the next 5 years, then probably everybody will have to go back to clinic once a year to get vaccination, which elderly people do against flu. So again, it shows that changes take place in the flu, and every year you need a new set of vaccines. So much we talked about immune system, so much we have talked about how developed our understanding of immunity is and how developed science is, and we are so good at technology. The irony is that even now, we don't have vaccine against many pathogens, viruses and bacteria, okay? In fact, we have no vaccinations against fungal infectious agent, and we have no vaccination against parasites. So look at the morbidity and mortality. Malaria, almost 500 million people suffer from malaria. Schistosomiasis, 15,000 deaths. I think it's almost 100,000 suffering one infection. Almost 1 billion people suffer from some form of one infection. They will never get rid of those worms throughout their life, and it will affect their quality of life all the time. Tuberculosis, so many people mortality, almost 2 million people die every year. AIDS, we'll talk about it today. Still there is no vaccine. Malaria, still there is no successful vaccine made in last 100 years or 120 years in the development of immunobiology. We still don't have vaccines against majority of the pathogens. And that is something which is a very burning question. We need to address them. We talked about active immunization. So you are actively challenging the immune system to get prepared through memory B cells and T cells. But there are also passive immunization. Once you have got the infection, then you need to treat people. And diphtheria and tetanus, we talked about their therapeutic approaches. So you take a container of tube that has already got antibodies against those components. So those antibodies will go and neutralize the pathogen. Very similar to early days of SARS-CoV-2 pandemic, where people were saying that if you take convalescent patient serum, people who have recovered from the infection, they should have antibodies which will protect you against subsequent infection in others. And that approach is called passive immunization. Can you use pre-made antibodies from natural source to stop infection in somebody who is infected now? Okay, so that's what you do. If there is a dog bite, then you go for rabies immunization, and that is therapeutic because you are getting antibodies which are already present from certain source, which are going to protect you. Same is true for hepatitis B and hepatitis B. If you think that you never had vaccination and you fear that you are going to have, you have exposed yourself to this kind of infectious agent, then there are opportunities for you to go to clinic or specialist shops where you can get these antibodies, and they're called passive immunization. Passive immunization is your immune system is not working to generate that immune response. The end product is already there, antibodies from different sources. So remember World War I, before penicillin came, people used to take pathogens, raise antibodies in horse, and then horse sera used to be used to treat people with serious infection. But such people also used to get what we call a serum sickness, and that was because of immune complex formation, type 3 hypersensitivity reaction. So these are the ways in which you can generate passive immunization. Nowadays, you can also use monoclonal antibodies. Okay. What are the safety problems with vaccinations? If you look at on the vaccine side, it has happened in the past, and when polio vaccine was being tested in on American Army, then there was instances of polio vaccine reverting back to wild type. They were attenuated, but they went back to become wild type, and people had polio because of vaccination. We are talking about 60s and 70s. So the place where I did my NASA fellowship in Salk Institute is named after Jonas Salk, who was the discoverer of polio vaccine. He made so much money that he decided to build that Salk Institute for Biological Studies in San Diego, California, and which is still the best private institution for research in America. So whatever he earned, he didn't keep it anything for himself. He just put it back into institute. Francis Crick, the double helix structure person who got Nobel Prize with Watson, he was the president of Salk Institute and my host for NASA fellowship. But he had instances where Jonas Salk found that a number of people had reversals of wild type. But that problem was sorted out later in 80s and 90s by developing better strategies to attenuate organisms, including polio. Sometimes killed organisms are not properly killed, especially heat killed. The Indian vaccine, which is being given, I think it is heat killed. But these are very specialized, evolved times. But historically, there are instances where the killing was not good enough, and there were enough polio pathogen left behind, which caused the polio disease instead of giving protective immune response. Many times the toxic material, like from typhoid or pertussis, that themselves can give toxic response in people. But these are very, very rare. You may be hearing a lot of stories about one person having this kind of immune response, and the other person was having very high fever, the other person had tremors, etc. These are very rare occasions because different people have different ways of coping with heightened immune response. So these are more exceptions than common events. When people used to attenuate them through animal cells, then animal themselves provided sources of animal viruses, and those contaminations 20, 30 years. Some people are hypersensitive to egg proteins, and some vaccines used to contain egg proteins to stabilize the vaccine components. So such people can have hypersensitive reaction because of the adjuvants or stabilizer added to the vaccine. Some people can have cross-reactivity, and that can lead to autoimmunity, rarest of rare incidents, but immunologically, these are possibilities. What about the patient side? It is very important to find out if the person who is being given the vaccine, especially attenuated vaccine, is immunocompetent, has normal immune system, not genetically inherited immune deficiency or not secondary immune deficiency, for example, infected with HIV-1. Adjuvants can trigger local inflammatory reactions because I said that a number of vaccines will have adjuvants which are designed to mimic PAMPs of pathogens. And that's why some people have swelling, some people have high fever because of TNF-alpha production. But eventually this will all taper down. It will just go away after a few hours, few days, or a week. Many times, some people who are hyper-excitable in terms of their immune system, they seem to develop immunopathology. But again, this is a rare case where the vaccine itself generates so much immune response. And you may have heard, one or two stories about SARS-CoV-2 vaccine as well, that people have overreacted. There is nothing unusual about it because this pathogen or the spike has not been seen before by our immune system. So some people are responding more dramatically, but it's more of an exception than the rule. A lot of people can be hypersensitive to the vaccine itself. Tetanus is one example. People take it and then they develop hypersensitivity reaction. Sometimes They go into mild anaphylaxis, but then they recover. Some vaccines are proposed to interfere with each other, but there is no way to prove it. There is no way to disprove it. And that's why most of the vaccines are spaced out quite conveniently so that you don't have all together. There are different months. So the immune system has opportunity to recover from the first vaccination and then prepare for the second vaccination. Some people have more heightened immune response. Respiratory syncytial virus is one example. Dengue is one example. And the most important thing, which we'll talk in the second half of today's talk, is about therapeutic vaccine. HIV-1 is a classical example. What about people who are already suffering from the disease? For example, tuberculosis, leprosy, and HIV-1, who are chronic long-term infection. So the most of the strategies are designed to offer prophylaxis, isn't it? But what about looking after people, at least millions of people who are already down with the disease? And there are a number of strategies which are being tried, which we classify as therapeutic vaccines, vaccines designed to treat the infection. So we have prophylactic vaccines, vaccines in anticipation of infection, which is the commonest. Then you have passive vaccines, passive immunization, and you have therapeutic vaccines to treat already infected people. Okay? We'll take 5 minutes break, and then we'll talk about the star pathogen of the year, isn't it? SARS-CoV-2. Okay?
Yes, sir. Sure, sir.
Right. So we start.
Yes, sir.
Can you see the slides?
Yes, sir. We can see the slides. Is visible, sir?
Okay, excellent. So now we are going to talk about, in the context of host immune response and the infection, we are going to talk about SARS-CoV-2 that causes the disease called COVID-19. We have published few papers recently, and you're most welcome to look into it. Um, Without going into too much details, as you know that it's a hyperinflammatory response. And I think it's a great opportunity for you to revisit the knowledge you have acquired about immune system, innate immune system and adaptive immune system in last 4 to 5 weeks. So probably you know that SARS-CoV-2 genome, and the most important to think about the spike protein. And it's a single stranded RNA genome, and it has got number of proteins, probably you know, through different articles. And the most important aspect is the spike protein, the S protein, which forms a trimer on the surface, and that is the main protein that interacts with a receptor, ACE2 receptor on the host cell, and the co-receptor TIMPRSS2, which causes the cleavage of the protein, and that leads to internalization of the virus. Okay, so this is quite important. Not all cells express ACE2 receptor and TIMPRSS2, but those who express, they don't have to be in the lungs. Cells which will express the co-receptor and the receptor, they're most likely to be permissive to SARS-CoV-2 virus. So the ACE2 and TIMPRSS2 is both are expressed in respiratory airways. So you can say different cells. Their co-expression is found in proximal areas and distal airways. Quite interestingly, the nasal cavity has highest expression of these two receptors and co-receptors. And the high expression is also there in the ciliated goblet cells, the cells which will produce mucus, etc. In other words, it explains why a number of people, especially young people, carry the virus in their nasal secretions or in nasal cavities, but they still don't go down with the disease. And that's where it demarcates the level of innate immunity activity against the virus. And also, people who are more susceptible, a less critical amount of virus is able to cause respiratory infection. In others, the people are carrying the virus without any symptoms, and we call them asymptomatic carriers. So the expression level of the receptors and co-receptors and the potency of your innate immunity, because type 2 cells and Clara cells and nasal cavity cells, they all are secreting some form of innate immune components, including including peptide antibiotics, which we have talked about, cathelicidins, etc. And that's why there is some sort of a standoff in people who are highly incompetent or those who are young and people who are not that immunocompetent, and that can dictate the severity of the disease. So many people have the virus, they're going around spreading it, mostly young people, but they don't seem to show any symptoms. And that is not primarily, it's very hard to say at this stage, that is being dictated by the amount of receptor, co-receptor expression and the potency of innate immune aspects, especially humoral factors. Just to show that the receptor binding domain is clearly binding to ACE2, which is facilitated by the TIMP-RSS2. And there are two states. In one state, it is a low affinity interaction. And in one other stage, where it is a stand-up state, which is through the exposure of a receptor binding domain, it is high affinity interaction. Okay. So these interactions of this trimeric spike protein, which is again the main component of vaccine development, they are very important for post-pathogen interaction on the target cells. The one good advantage of the other stage is that the conformation of a spike protein will mask, and that will probably allow masking of those sites which are convenient targets of immune system, including innate immune factors. Not only your nasal cavity and respiratory airway cells, which you can see here, but there are non-respiratory cells as well, which express ACE2 and TIMP-RSS2, cornea, gallbladder, bile duct, hepatic duct, prostate, testis, esophagus, colon. And that probably explains why some people don't show any respiratory complications, but they show complications of heart or the complications of GIT, because some people have non-stop diarrhea, but they don't have any problems with the lungs. There is also another aspect that we talk as long COVID, that people who have recovered from the disease may have long-term complications of the infection, even if the virus has gone away. And that's simply because virus may have triggered post-immune response or immunopathology or metabolic changes because they bound to those target cells. And even if the virus has gone, because of constant exposure of virus with these ACE2 and cases of this infection, and only time will tell how it is going to affect. What we know from pregnancy is that in many countries, women who had infection with SARS-CoV-2 in the early part of their pregnancy, then they have recovered, they're perfectly fine. Then there was no problem in their second trimester and third trimester of pregnancy, but their placenta is very, very sick and babies are very small sized. But in some countries, there is no problem. For example, Chinese have reported that it doesn't affect pregnancy. In India, people have reported that mothers seem to develop more of preeclampsia, where there is high blood pressure in mothers, and they have more pressure on kidney, and they have proteinuria during pregnancy. So that can affect the gestational aspect of the baby. So there are different countries and different demographics are reporting different implications of long-term COVID, and slowly we'll figure out the impact of long-term COVID. One thing is quite clear, talking to some of the lung specialists or chest specialists, that many people seem to have symptoms of classical fibrosis in their lungs, even if they have recovered from the disease, even after 2-3 months since the virus has gone away. And that could all come down to a combination of immunopathology, that virus has actually triggered an immune response that is not, which is continued, which doesn't stop. There's a constitutive takeover immune response. And the second is that virus has damaged a number of cells during immunopathology. It is also possible that virus has become latent. We don't know in most cases. So it's a very intriguing pathogen. And when you know that when there's a new pathogen, it takes time to understand how it works, how it manages, how it engineers immune response, how it suppresses immune response, how it escapes from immune response. And this is a very nice diagram drawn by one of my PhDs, which is published in Journal Immunobiology, where it shows in the lungs that the virus, in the process of infection, what it does is to activate immune cells, which churn out a number of cytokines, mostly pro-inflammatory cytokines, cytokines like IL-1 beta, IL-6, and TNF-alpha. And we have talked about massive infiltration of immune cells. So your lung cavity is full of a number of cytokines, and TNF-alpha will cause vascular leakage, as you can see here. Then there'll be more neutrophils, polymorphonuclear cells getting infiltrated. And slowly, these lungs will have more mucus, more apoptotic cells. And that's the time when these lungs become so occupied with fluid material and cellular debris, cytokines. The more cytokines are produced, the more cellular infiltration will take place. The more neutrophils will come, which are terminally differentiated. Within 12 hours, they will die. So all this debris starts to accumulate, and that's why people start to have problem with breathing. But what is interesting to recognize is that the oxygen saturation in the periphery, that is in circulation, starts to go down dramatically before the lungs start to have low oxygen. So it was not a physical process or a pathophysiological process. And then people started to recognize or started to ask the question, is the virus elsewhere as well? Very few organs, very few groups have reported the existence of virus in different organs. In fact, people don't believe that the virus exists in blood. There is no viremia like HIV-1 or some other viruses. What virus does is to trigger the most important inflammatory component, that is complement system. And the complement system is involved in the development of all complications of COVID-19, as we'll talk about quite soon. So this highlights one of the pictures, figures probably done by one of our collaborators, Hadida. Activation of a number of immune cells, which we have discussed throughout last 5 weeks. And we have talked about cytokine storm, pro-inflammatory cytokine storm. And then there is activation of coagulation, and that leads to what we call as coagulopathy. Probably we haven't discussed it, but there's a direct link between complement activation and coagulation system. It is also possible that whenever there is a tissue damage, coagulation proteins get activated, and the cascade will lead to development of a clot. You should remember that the clot is seen as altered self by our complement system, especially C1q, and that will lead to more activation of complement and C5 operating cells. And that we call as vascular permeability alteration, which is microangiopathy, where vesicles, blood vessels, that trouble that can the side the and some people have problem with breathing. Hello?
Yes, sir.
Can you hear me? I
Yes, sir.
disappeared sometime.
Yes, sir.
Can you
Yes,
see
sir.
the
You're
slide?
audible now.
Where we are on this slide or before that?
No, sir. We are in this slide itself.
Was I talking about this slide
Yes,
or
sir.
the slide
Yes,
before?
sir. No, sir. This slide itself you were talking about.
All right, okay. So what I was saying is that is them pro-inflammatory cytokines. If they are not overproduced, then they will deal with the pathogen. Type 1 interferon is made that will degrade the virus. Most of the processes which we have talked about, they are all working fine against SARS-CoV-2. It's only when things go out of control, and that's where people started to find out or interrogate why people are having oxygen deficit in the periphery, and then people were going into coma and organ failure, etc. So the coagulopathy and antiphospholipid antibodies are quite rampant in COVID-19 patients, severe patients. And these severe COVID-19 patients also seem to have endothelial activation. And people who were admitted or who are continue to be admitted to intensive care unit, ICU units, they seem to have thrombotic complications, so blood infarcts, clots, etc., going everywhere. So obviously it refers to complement activation, coagulation, activation, hyperactivation, that is coagulopathy, and clots not getting dissolved or not getting removed. What was the evidence? The clinical evidence that came in March last year, in 2020, is that there was systemic thrombosis in these patients, and that is referring to coagulation dysfunction. So people were not able to coagulate properly, and that is measured by high levels of D dimers, LDH, and bilirubin, platelets, low levels. And then people started to notice that there was damage to kidney, and a lot of people were then immediately they had to go on ventilators. The curious thing is that although there was massive fluid accumulation in the lungs for reasons we have talked about, there was no connection between and cardiac injury. And then people found that there was microvascular thrombi in various organs as well when people started to look at autopsies starting from Wuhan in China. So people who have died, they had lots of thrombi, plaques, and clots all over different organs. Again, referring to hyperactivation of complement, microangiopathy, and problem with the clot formation. And that's why people have tried to use complement inhibitors to stop this process of thrombotic microangiopathy, cardiac dysfunctions, etc. Because an antibody which can interfere with C5a, it won't let C5a generate, so anaphylactic processes are recovered, and that has worked in the past in one disease called hemolytic uremic syndrome. So people have tried to stop that organ recovery, and there have been clinical trials where anti-C5 antibodies have stopped people from entering into thrombotic microangiopathy in COVID-19 severe cases. Then the most important aspect is that these are the therapeutic aspects. What about the vaccines? Now you know that there are RNA-based vaccines. So there were almost 180 candidates a year ago, and now we have come down to 6 or 8, which are actually being given to people. And that tells you that how many ideas actually get translated to the clinic. And the most important things which you need to remember is that there is a protein-based epitope-based vaccine from Imperial College, the RNA vaccine, which Pfizer and biotech NT is BioNTech is giving. Then there are DNA vaccines. There's an inactivated adenoviral inactivated virus vaccine. So the one from Hyderabad, this one, and then there is the adenoviral scaffold, the chimp flu adenovirus vaccine is from Oxford, that was made by AstraZeneca, and Serum Institute in Pune is also repackaging and recycling it. And the one which is coming from China is the human adenoviral vector containing the spike protein. The whole idea is that once these vaccines go in, then macrophages or dendritic cells will grab them, and then they will present it to T helper cells. Th2 cytokines will ask B cells to make antibodies. These antibodies, these things have been tested in animals. And these antibodies they can present these HIV-1 cis or trans to T cell, and then infection will proceed. So these pathogens, HIV-1, is very smart in using the antigen presenting cells as their vehicles to reach T cells when it is required. So many times you have killed all the T cells, you have got rid of all the extracellular viruses. There can be macrophages and DCs which can continue to carry HIV-1 inside them. Very convenient carriers for them. And it is that time when the clinical latency period is breached, then you have full-blown AIDS, which we talked about in previous slides. Can you still hear me?
Yes, sir.
Yes, sir, you're
Excellent.
audible.
One of the issues is that the antibodies which are being made during this clinical phase, latency phase, you can see here, antibodies are made, cytotoxic T cell response continues, that is CD8 positive cells. And during these constitutive periods, the antibodies which are being made, they continue to neutralize. But when the set point is breached, then you can see the virus is going up in spite of antibodies against P24, antibodies against GP120, etc. Why is that happening? Now, here is the problem with HIV-1, and that's why we don't have a vaccine in spite of 30 years of research and very serious research. A number of antibodies which we make, they are now neutralizing antibodies. Okay, so they are not able to inhibit HIV-1 from binding to CD4 and the chemokine receptor, so they can still infect. We talked about the set point, but once virus set point is achieved, the CD8 cells, that is cytotoxic T cells, will continue to suppress HIV-1. But there is a downside. If the HIV-1 seems to think that, okay, how long can I persist as set point? I need to do something. That is the time when this virus starts to make mutations, because there is an immune pressure on the virus, okay? And because of that set point which has achieved, what we see as host pathogen standoff is basically an opportunity for the virus to create new variants, new mutants. And you talk about these new variants coming out, even with SARS-CoV-2, they appear simply because they need to change, because more and more people start to produce good quality antibodies against them. So there is an immune pressure on the pathogen to change. So in an attempt to protect ourselves, we are also forcing these pathogens to change. If we don't put immune pressure on them, they won't change. They will keep on infecting and surviving. That's what they want. But that will lead to generation of mutants. Okay? And these mutations take place in GP41. There's a structure called loop structure. So what happens is that, for example, if you have made antibody against HIV-1, then in 2-3 months' time, that same HIV-1 will change to HIV-1B instead of A. Then obviously HIV-1A antibody is not going to neutralize B pathogen. By the time immune cells start to mount immune response against B, the antibodies are there in 3 to 4 weeks. It has become C. In other words, our immune system or humoral factors involving antibodies are constantly changing the host. Anti-A, by the time it is ready, anti-A virus doesn't exist. It is the B. Then anti-B comes, then it has become C. It is very similar to what happens in a number of parasites. They keep on changing their virulence proteins. Okay? So in other words, what I have been saying is that variants emerge throughout this set point, and these variants are quite capable of infecting because there is no fitness cost. Mutations it does make in the loop or hypervariabilism in the GP120, but it doesn't affect their capability or capacity to infect CD4 positive T cells. So they continue to infect. And that's why after the set point is breached, because of the number or accumulation of mutants, what we call as escape mutants which have escaped from immune assault, they breach the set point and then cause the full-blown disease. What is the expectation? The same question was asked 30 years ago. We continue to ask this question. Okay. We need a neutralizing antibody vaccine, a vaccine that is neutralizing, which doesn't allow virus to infect. We also need a vaccine that will stimulate T cell response, because we want to stimulate CD4 positive T cells. Otherwise, antibody production will not take place. And what about therapeutic vaccine? Those 50 million people who are already suffering from HIV-1, they are constantly carrying the virus throughout their life in spite of these antiretroviral drugs, which will just keep them suppressed. We need vaccines so that their infection can be completely removed. Now there are two serious problems with this virus, HIV-1, which was realized a bit late. The virus, when it binds to the target cell, it can rotate on its axis. Okay, so it changes conformation. So number it generates escapes in GP120, and then it can rotate on its axis. The most important aspect, we have talked about cell surface is coated with sialic acid, and that sialic acid is a signature of self. What this virus does after binding to cell or while it is interacting, it imports sialic acid on its surface in an attempt to fool our immune system that it is not non-self, it is actually self, because it gets coated with sialic acids, carbohydrates here. And in that case, the immune system is becoming more permissive. It allows you to interact with T cells, thinking that it is a self-signature containing molecule or protein, and that's how it escapes. The other problem with vaccine is very complicated. So there are not one type of HIV-1. There are three types of HIV-1, M, N, O, depending on the sequences. So these are different. And within, for example, one type, which is the main M type, there are further subtypes. We call them clades, amino acid sequences differences. And then there are A, B, C, D, E, G clades. Each clade will differ by nearly 20% in sequence. Within each clade, there is up to 10% sequence variability. So first you have main type, then subtypes, that is clade. Each clade from each other differ by 20%. So 1/5 is of A is different from B, so on and so forth. And within A, there is 10% sequence variability. So if you have M strain or type with A subtype, you may have a number of viruses with M and A combination, but A can have a lot of differences from other M and A. So there are lots of sequence variabilities within this. This will add to complications in generating escape mutations. How? Now here you look. You have B here, the clade in America, in South America, in Africa you have A, D, and C. If you go further North Africa, you will see G. If you come to India, it is C. This part, C. If you go to Australia, it is B. So these are the strains with clades which are rampant in these parts of the world. So you can't have a universal vaccine against HIV-1, because the vaccine which is going to work in America, North America, and South America is unlikely to work in India, right? Because there is so much variability of clades and sequence, so they are not able to neutralize. So what is going to work here in America is going to work in Australia, but not here, very neighboring country. Okay. And these are the complications which makes it very difficult. What many people have done, including the group in Oxford who are our neighbors, they spent 20 years generating these vaccines. They took all the sequences from each clade to generate an universal vaccine. So all those sequence variabilities were cloned together like a spike protein and then challenged to SIV1. The irony is that every HIV-1 vaccine works beautifully in monkeys.
Давай ты, Jason. Kita mau Jason. Kau boleh. Wait.
I'm present over here. So, what I wanted to, am I audible?
Yes, yes, you're very audible.
Okay. So, sir, what I wanted to ask was that, so why exactly does a foreign molecule need to be non-potent only to be able to qualify to be the antigen causing hypersensitivity? What is the whole story around it being non-potent?
I don't know what you mean by non-potent. So if you are atopic, then or genetically predisposed, then what is normal antigen for others becomes allergen for you. For example, if I come across house dust mite allergen like DERP1, I will make an IgG antibodies and it will neutralize it. But somebody who is atopic, genetically predisposed, will make more IL-13 in response, Th2 response, and you'll make more IgE type antibodies. So the moment context changes, IgE antibodies will go and bind to mast cells and basophils, and then allergen hypersensitivity reaction can set in. So that decision is taken by your genetic susceptibility and what cytokine you make. And that's why by priming at root changes, you induce your immune system to make more IgG antibodies, which we describe as immunotherapy. So there are peptides which are very innocuous for people who are not allergic. We call them non-atopic. And there are peptides and proteins. Some people respond overwhelmingly simply because they have MSC predisposition to that. Does that answer your question?
Yes, sir. That leads me to another question. If we go to the root of all of this, would we ultimately infer that genetic predisposition would be the main role player in terms of the whole picture of hypersensitivity?
That's true with almost everything. You can always blame your parents and grandparents for this.
Okay,
It's true for
sir.
everything. The genes you inherit can dictate your susceptibility, isn't it? But
Right.
yes, there are environmental factors. Nowadays, there are microbiome that how much microbiome mothers pass on to baby can dictate the level of colitis and milk tolerance, rather lactose tolerance, and their susceptibility to developing autoimmune disease. Uh, but this goes on, isn't it? Ultimately, it's a combination of genetic and epigenetic factors, the environmental factors, which affect, for example, multiple sclerosis is very common in the Western world, but in India, you don't have that problem. It's not that HLA-DR molecules are not rampant. So is it the food quality or there are other genetic factors which we have in India which don't allow MS to be a problem, isn't it? Rheumatoid arthritis is rampant in India, isn't it? Asthma and fibrosis are rampant. I can understand it because big cities are very polluted. But I'm sure that there are elements of genetics that makes you more susceptible to asthma. Because here also, I think it's a clean air, but it's not that asthma is quite less. So I think it's a combination of both. It's an empirical question. You just don't know which one is contributing to how much, isn't it?
Yes. So I would like to follow up with one more question. If a person does not have any form of, like if the genome of a person does not have any form of implication in the whole scene, then would a person still manifest some form of hypersensitivity in his lifetime, his or her lifetime?
We don't know. I mean, it's very hard to sequence the genome and predict anything, right?
Right.
So if you go on to develop hypersensitivity, you can argue that it was epigenetic factor. So it was less of genetics and more of environmental factors, isn't it? So ultimately, what
Right,
skews
sir.
the balance towards hypersensitivity, we don't know. It's a T cell polarization, isn't it? Same is true for autoimmunity. If you look at why somebody is making antibodies against, say, acetylcholine receptor, or